Hello everyone, this is Mr. Rossi. I hope you guys are all doing okay at home, not going too crazy, and you're healthy, and your whole family's doing pretty well right now. I know I'm going crazy. Hope you guys are not. I'm currently in my garage trying to hide from my children to do this little video for you for the first time, so let's see how it goes. Alright, so our next unit here is going to be involving mitosis and meiosis. Pretty much the idea is how a cell can divide, either to simply copy itself, or when the need comes that you want to reproduce, that you're going to produce a sperm or an egg cell, depending on if you're male or female then, obviously, at that point. So today's lecture is just going to be about the concept of what we call the cell cycle. Alright, now what the cell cycle is pretty much the period of growth and development of a very specific type of cell. And that cell is going to be called a somatic cell. So that's what we're going to be starting with today in a moment. So as I proceed here through into the PowerPoint, we're going to start talking about this. So first, somatic cell. All right, and that's what we're going to be involved with today for the cell cycle, as well as eventually mitosis, which will be the next lecture I provide for you. A somatic cell pretty much refers to any cell in your entire body. Almost any cell in your entire body. Alright, so this is going to be anything pretty much except a sperm or an egg cell. So skin cells, heart cells, brain cells, you name it, they're considered somatic. And if you're human, obviously we all are, but you could be some other type of organism on Earth, but if you're human, every somatic cell is going to have a total of 46 chromosomes. I know we've gone over this 100 times in class before we left for school for the COVID crisis that we're having now, so this shouldn't be too much of a shock to you. So again, our focus right now is going to be on somatic cells, all right? Body cells that have, if you're human, a total of 46 chromosomes. When we get to meiosis, we're going to be focusing on what we call gametes, or sex cells. Gamete is like the official term for it. All right, so a gamete or sex cell will be a sperm or an egg, and they're going to have 23 chromosomes, again, if you're human. The idea being that you're only going to provide half of your overall genetic information to your child one day, and then your spouse would provide the other. So 23 from one egg, 23 from one sperm, put them together after fertilization, you have a cell that would now be considered somatic with a total of 46 chromosomes. So those are our two designations. So why are we really even talking about this in the first place? Well, you got to understand, we are an organism that is made up of roughly at least 1 trillion cells. We're not just one big, gross blob cell surviving independently. All right? We are one individual or one organism that's actually made up of 1 trillion cells that are all working together in tandem, maintaining what we talked about in the very first week of school, homeostasis. All right, so eventually, though, cells age, just like you and I are aging every day and every year. All right, as they get old, they don't function as well anymore, or they die. So the idea being is you would want to replace these old, worn out, or dying cells before you lose them forever. That way you can always have every tissue, every organ, everything in your entire body working at hopefully at 100% efficiency or capacity, maintaining homeostasis. So the next slide we're really going to dive into is, again, why do cells ultimately have to divide? All right, this again is just a picture giving you an idea that these are all somatic cells here: brain, liver, muscle, blood, intestine. Again, anything besides sperm or egg, which we consider gametes. All right, so cell growth and limiting factors. So ultimately, again, why is cell size limited? Why are we not all this big Bob here from the movie Monsters vs. Aliens, if you've ever seen it? Why don't we look like this? Why are we just not one big gross cell? Well, I kind of already hinted at it, that if you were to die, then you're gone forever. All right, but that's really not the ultimate reason why we're not just one big Bob here. The ultimate reason is the rate of diffusion. All right, as a cell gets bigger and bigger and bigger, its rate of diffusion is going to decrease. And the bigger it gets, its rate of diffusion is actually going to decrease kind of more rapidly as it goes. So if you're a big gigantic cell like Bob and your diffusion rate starts to decrease, what that means is you're not going to be able to get rid of waste out of your cell fast enough or get the materials that your cell needs to the correct location fast enough. So think of it like this. You have to go to the bathroom and you've been holding it for the last 2 hours. You feel like your bladder is going to explode. Well, if you held it long enough, theoretically it could and release all those toxins into your entire body, and you can get a septic shock or get a bacterial infection in your body. Well, the same thing if you're a really big, huge cell and you can't get rid of your waste properly fast enough through diffusion, you're also going to suffer and get sick and die. All right. Also, though, the rate of protein that you're producing is going to decrease. The amount of food that you can bring into the cell and get to where it needs to go is going to decrease. So it's not good to be gigantic. All right, and again, the larger a cell's volume, so how much can fit inside of it, right, the slower its rate of diffusion will be, and that cell is going to suffer. So what have we decided to do? We've become multicellular. As I said, we're made of 1 trillion cells. So we are made of 1 trillion cells that are all functioning together, but ultimately they're really small, so their rate of diffusion never is drastically impacted on. So if we look at a diagram like this, here is a very large cell, and if we calculated its surface area, it looks really big, right? It's 5,400 microns squared, a measurement for a microscopic value. All right, and that seems pretty big, right? But this cell here would suffer drastically due to its overall volume compared to its surface area. It's too big to get things in or out of it fast enough, especially here from the nucleus. But if you create a bunch of cells, smaller cells, connect them together to make them multicellular, they're pretty much, it looks like taking up the same amount of space. But now look, the surface area is actually drastically larger, 16,200 micrometers squared. But the volume of each individual cell is much smaller. So they're not going to have the issue with diffusion, things going in or out, as much as one big gigantic cell like Bob would be. So that's the advantage here. So our rate of diffusion does not get too slow. That is going to impact the cell's ability to survive. Now you're going to work on a worksheet for me that's going to be due on the next day by 2:20 p.m. So this idea here again, small cell, if you were to calculate its surface area, length times width times it has 6 sides. So length and width obviously are 1 centimeter. So you do 1 times 1 times 6 sides. It gives you 6 centimeters squared is the surface area. If you were to look at a big, huge, gigantic cell like you see over here, 3 times 3 times 6 sides again gives you 54 centimeters squared. So the surface area of this cell here is much bigger than it is for this small cell here. But that's not what we're really ultimately worried about. We're worried about what the volume is going to be. So for the smaller cell, my volume is going to be length times width times height. So that's some super easy math. 1 times 1 times 1 is going to be 1 centimeter cubed. We have to keep track of how many centimeters here we're multiplying, so it's cubed. This guy over here, though, 3 times 3 times 3 gives me 27 centimeters cubed, a much larger volume. Ultimately, what we want to look at, though, is something called the ratio of surface area to volume. All right, you want to have a large surface area combined to a smaller volume. So for this small cell, my surface area to volume ratio is 6 to 1. All right, so again, I have 6 surface area, which is larger compared to my overall volume. But as you get bigger, so if you look at this cell and then eventually the one we were talking about earlier, now look at it. Surface area 54 centimeters squared, volume 27 centimeters cubed, 54 to 27, or if I reduce that, I get a ratio of 2 to 1. Now, the closer that ratio is, all right, 2 to 1 or 3 to 1 over here for this one here that's in the middle, all right, that's not good compared to a very large surface area, the small volume ratio here. So this is the type of ratio you'd be looking for a cell to make sure it's functioning adequately. As that cell gets bigger, its surface area to volume ratio is always going to decrease. All right, eventually if it gets too close to 1-to-1, that cell is suffering, it can't maintain diffusion properly, it's starving, it's going to die. All right, so your worksheet that you're going to get assigned, you're going to have to make some of these calculations, so you might want to refer back to this video here. All right, now moving forward, all right, we're going to go through why do cells divide besides getting too big in the first place. So maybe they're just too old, so they want to be replaced. Maybe they become damaged, you got a little paper cut and you want to fix them and seal that wound. If you are an adolescent and you're currently growing, you've entered puberty, you're adding more and more cells so you can get bigger. Or eventually, if you want to reproduce, you're going to make sperm or egg as well. So there's lots of other reasons that cells are going to divide besides they're just getting too big in general. So, chromatin, chromosomes, and chromatids. Now, these are terms we're familiar with to a degree. We're familiar with chromatin, which is what we said before is the form of DNA when a cell is dividing. So that's this stuff down here. Looks like a big bowl of spaghetti or lo mein. Now, you're going to see chromosomes. They're going to look like thick rods like this once a cell is getting ready to divide. All right, so once the cell is going to enter division, all of the chromatin will condense to form as many chromosomes as it has in particular, and we have 46 total. Now, before a cell divides, it's going to replicate its DNA. So if we look at this down here, all right, this says homologous chromosomes. Prefix homo means the same. So we got two chromosomes here that really have the same type of information. However, before a cell divides, you're going to want to copy each of these two chromosomes. So by the time cell division occurs, the chromosomes are actually going to look like this. They're going to look like X's here. All right. Now, what you did was you took one chromosome and copied it. So this X right here is pretty much two identical chromosomes connected in the middle by what we call a centromere. All right. When I look at this X right here, as long as the two halves here are connected by a centromere, I'm going to call this one chromosome. Or I now have two chromatids. I'm pointing to these sides of the X over here. And a chromatid, again, is simply a copy of a chromosome. Or when the two chromatids are connected together here, we're going to call it a sister chromatid pair. Two chromatids connected in the middle by a centromere. So if I just blow this picture up again, here is right now 2 chromosomes. Here's your original 2 chromosomes if you only had 2. By the time division begins, your 2 chromosomes are going to look like this, 2 X's. You still count the centromeres, so you have 2 chromosomes. But now each chromosome is consisting of 2 chromatids, 2 halves of the X, or 1 sister chromatid pair as long as they're still connected. So right now I have 2 chromosomes, or 2 sister chromatid pairs, or a total of 1, 2, 3, 4 chromatids. Now these terms get a little bit confusing when we move on to mitosis, but we simply got to remember that chromosomes are going to copy themselves before they can go through division. So each chromosome will look like an X. Each X comprises of 2 chromatids. I still would just call it 1 chromosome until they separate, or 1 sister chromatid pair.